So again, like usual, if you have any questions or if I'm saying anything wrong, feel free to correct me and put it in chat. And yeah, okay. So 11.1 is antibody production and vaccines. And this is all about basically immunity and immunology. But yeah. So the first thing that we need to know is about surfaces of cells. So all living organisms, whether that's humans, whether that's other microorganisms, have proteins and other substances on the surface of their cells, and specifically proteins. So some organisms have a cell wall outside their plasma membranes, which are made of polysaccharides or other substances. And those are usually in the cases of certain prokaryotes or plant cells. So usually on our plasma membranes, there are proteins that do essential work for us, basically. Viruses, on the other hand, are not considered living organisms and are not composed of any cells, but they also have unique molecules on their surface. So the surface of most viruses is a protein coat called capsid, which basically is similar to what we have in terms of plasma membranes, but they have the capsid, which is a protein coat, basically. So the capsid of some viruses is enveloped in a membrane that is taken from the plasma membrane of the host cell. So when viruses take over our body, for example, and they took over a certain host cell, the plasma membrane of our host cell will be the capsid, will envelop the capsid of of the virus that's affecting us. So the unique surface molecules in both in living organisms or in viruses are used in several ways. So in this in the context of viruses recognize and bind to their host using molecules on the surface of their host cells. So when viruses enter our body, the capsid will basically allow them to recognize our host cells, our cells, which are for them the host cells, and therefore take over the host cell. In living organisms, there's two main functions. Firstly, it recognizes its own cells and cell types using surface molecules, so that you know that the cell that you are recognizing is your own. But it also recognizes pathogenic cells and viruses by the surface molecules that are not present in our organism, in the organism itself or in us. So they're foreign substances. So these molecules will trigger the production of antibodies. So they're called antigens. So when certain living organisms recognize the surface molecules on pathogenic cells and viruses as foreign, the surface molecules on the pathogen are called antigens because they trigger antibody production within our cells. When we learn about antigens, we learn about antigens on blood cells. So all blood cells have antigens on them. And the ABO blood group system is based on the presence or absence of glycoproteins in the membranes of our red blood cells. So the glycoproteins that are on our red blood cells are what determine whether or not you are type A, type B, or type O, or type AB. So those glycoproteins can cause antibody production if a person doesn't naturally have them. So for example, if I'm type O, so if I receive type A blood, I will see there will be it will trigger antibody production within my body because there's no naturally existing type A or type B glycoprotein. So all the glycoproteins on the surface of red blood cells are known as antigens because of this. That's why we call them antigens on blood cells. So O, A, and B are the three types of antigens of the basically O, A, and B antigens are the three versions of the glycoprotein. So this is what they look like. O antigen over here is just the basic square in this, sorry, a diamond in this case. And then the O antigen is present in O, A, or AB. But the A antigen is made by adding N-acetyl galactosamine molecule. I can't pronounce that correctly ever, but basically this specific molecule is added to the O antigen, which is this dark gray color molecule here. This is added to the O antigen, and then this is what makes the A antigen. The B antigen works in a similar way, but by adding another galactose, it creates the B antigen. Adding a galactose to the already existing O glycoprotein will create the B antigen. So therefore, in blood group O, the only antigen that's naturally present is O. But in A, B, and AB, all three are in A, B, and AB, O is present in all three, which is why you consider the O group to be very good in terms of being able to be a good donor. Of course, the O positive and O negative will cause problems, but we don't learn about those here. So basically, if you're the O blood group, you can only receive type O blood because type A, type B, or type AB blood will have A or B antigens, and that will therefore cause basically an immune response against the blood. In type A, it's just the antigen B or type AB blood, and in blood group B, O and B exist, so A or AB type blood or just the antigen A is what causes antibody production. However, in type AB, it has all O, A, and B antigens present, which means that there's no type of blood or no antigen that can cause antibody production, which is why AB is considered really, basically AB plus or minus, if we disregard the plus or minus, AB is a really good recipient. Okay, the next is histamine and allergies. I don't remember, who wanted to know about histamine? Are they here? Whoever, I swear there was someone who said ping me when we talk about histamine. But there's just so much in chat now. Okay. It was, yeah. I'm just gonna ping them anyway. I think they're here already, but I'm just gonna ping them anyway. Histamines. I say in all caps. Okay. I'm just gonna wait for a second, just so whoever it was can come and watch or listen. Okay, I'll just go into it anyway, just because we're gonna run out of time if we wait longer, but I hope they'll make their way here. Anyway, so there's two types of cell in our body which secrete histamine. There's the basophils and the mast cells. So both of these are types of white blood cells, and basophils are a type of white blood cell, but mast cells are similar to basophils but are found in the connective tissue. Histamine is secreted in response to local infections, and basically histamine causes vasodilation, which means dilation of capillaries or any small blood vessels in the infected area. The vessels become quote unquote leaky. Basically, they increase the flow of fluid that contains white blood cells to the infected area to be able to to be able to treat the infection, essentially. Allergies are reactions by the immune system to substances in the environment that are normally harmless. So, pollen, bee stings, or specific foods such as peanuts are usually harmless substances, but people who are allergic to these substances um, basically have an immune system reaction to them. So, substances in these allergens cause an overactivation of basophils and mast cells, which are obviously the two that secrete histamine, and therefore excessive secretion of histamine. This causes the symptoms that are associated with our allergies, so inflammation of tissues, itching, mucus secretion, and sneezing are the four symptoms, and it's caused because of the excessive secretion of histamine. Histamine is also implicated in the formation of allergic rashes. So if people break out into hives as an allergic reaction, or swelling known as anaphylaxis, when inflammation occurs, but it occurs in your trachea, so it blocks your airway. That's anaphylaxis. That occurs because of histamines. So to lessen the effects of allergic responses, the drugs we take are antihistamines. So any anti-allergic medication is an antihistamine. Are there any questions so far before I go into what is a local infection? So an infection usually occurs in one site, right? So for example, if you get strep throat, the infection is localized to your throat, or localized to your trachea or near your trachea, so that's a local infection. Sometimes you can have sepsis, which means the infection has seeped into your blood. Histamines don't really act on those. We've covered today 6.6, and then we're just going into 11.1. Why specifically do small blood cells get dilated? Because since they're local infections, there's not really big blood cells don't get dilated, blood vessels get dilated. When you have local infections, you won't have any big vessels nearby. So it's usually broken down into either capillaries or arterioles, and then those will dilate because there's no big vessels nearby. That's just because of how our body is made up. It doesn't have to be. Yes. Yes. Your immune system looks at dust mites to be a pathogen, and therefore will react to it to trigger the basophils and mast cells to produce histamine. What if you have histamine allergy? I don't think people have histamine allergies, but I don't think I'm even qualified to answer that question in the first place anyway. Because histamine is an internal thing, right? Your body creates it on its own. I don't think it's very rare for you to have an allergic reaction to your own body thing, but if that occurs, that's usually what autoimmune conditions are, and that's why you need treatment usually. But I don't know of that case, so I can't say. Histamine is the one signaling that you have an allergic reaction. Basically, histamine is the one that causes the reaction to take place in the first place. So your immune system will regard allergens. This is HLES. So your immune system will look at allergens as as if they're pathogens, and then look at it as if it's a local infection, and then basophils and mast cells will be triggered, and then the histamine will be produced. So histamine is not the one signaling that there's an allergic reaction, but histamine is the one that basically causes the reaction to happen in the first place. The only SL thing I did today was 6.6, and I'll go through option C later on if you I mean I'll ping everyone when I do option C if you care about that at all. And if you're looking for more SL stuff, if you go into I don't know if they're still here and I'm just talking to no one, but if you want to look at more SL stuff, you can go into the M24 bio chat and look at pins, and there's a bunch of recordings from old sessions that we did, and those are SL specific. This is 11.1. Okay, so stages of antibiotic production. So there's 4 stages. The first is activation of helper T cells. So helper T cells have antibody-like receptor proteins in their plasma membrane to which one specific antigen can bind. So if you recall back to when we did 6.3, I want to say yesterday, antibodies are pathogen specific or antigen specific. So helper T cells basically have receptor proteins on their membrane that are similar to antibodies, and only one specific antigen can bind to it. When the antigen binds to it, the helper T cell is activated. So if you look over here, it's an inactive helper cell. This is a receptor protein that's similar to the antibody. And then the antigen is presented to it by a macrophage, which is a phagocytic white blood cell. The macrophage will bring the so if you remember phagocytes from yesterday, those will bring the antigens, present it to the T cell, and then activate it. And then the second step is activation of B cells. So inactive B cells have antibodies. So this, for example, is an inactive B cell, and they have this antibody. So they have antibodies in their plasma membrane, and then if these antibodies match an antigen, the antigen will bind to the antibody. However, they're not active yet. So an activated helper T cell with the receptors for the same antigen, so the helper T cell that was activated here will then bind to the B cell and then send a signal to the B cell to activate it. So if you want a visual, basically it'll start with an inactive T cell which gets activated by an antigen which is presented to it by a macrophage. After it gets activated, the same antigen binds to the antibody in the membrane of a B cell that's nearby, but this B cell remains inactive. Then the activated helper T cell from here will bind to the B cell to enclose the antigen, and then will send a signal to activate this B cell to close it, to do the next action. But basically it will close it in between the antibody-like receptor and the actual antibody. And then, sorry, this is supposed to say 3 and 4. That's my bad. 3. And then 4. Okay. So, production of plasma cells is after B cells have been activated. So activated B cells will divide by mitosis to form a clone of cells. These cells become active, which with a much greater volume of cytoplasm, and then these active cells will together be called as the plasma cells. They have a very extensive network of RER, and they're used for synthesis of large amounts of antibodies, which is then secreted by exocytosis. So basically, this cell, after being activated, will have a large amount of rough endoplasmic reticulum, which will synthesize a lot of protein antibodies, because antibodies are basically nothing but globular proteins. So those antibodies will be produced by the rough endoplasmic reticulum in the plasma cells, because this B cell will divide into plasma cells. The plasma cells have a lot of endoplasmic reticulum, and then they will synthesize large amounts of antibodies, which is then secreted by the exocytosis. And then those antibodies will basically disintegrate the antigen and then kill it. And then the fourth step of antibiotic production is the production of memory cells. So memory cells are basically B cells and T cells that are formed at the same time as activated helper T cells and B cells when a disease challenges the immune system. But after the activated ones are gone and the antibodies that are produced have disappeared, these memory cells will still exist and allow a rapid response if the disease is encountered again. So memory cells will give a long-term immunity to a disease. Is there any questions with that? Sorry, antibodies. Yes, it means antibodies. It's stages of antibody production. I will change that. Antibody production. Okay. Yeah. Yes. And then that antigen is presented to the T cell, which then activates it. But then that antigen will not stay bound to it, but then instead will stay bound to a B cell, which then the activated T cell will help will bind to the B cell and connect it, which will therefore trigger the inactive B cell to turn into an activated B cell. Which one was the one that engulfed and died fast, displaying the antibody of the pathogen? I don't understand that question. I think you mean phagocytes. A macrophage is basically a phagocytic white blood cells, which basically means that it engulfs the antigen or the pathogen and through endocytosis. There's phagocytes, and then the other ones that engulfed and committed suicide. Lysosomes? Lysozymes? Do you mean lysozymes? I don't know. Wait, I might be... Hold on. Let me just check this. Because I just remember phagocytes and then... Oh, I know what you're talking about. Hold on. We literally did this yesterday. I don't know why I'm struggling. It's, hold on. Lymphocytes. Sorry. Lymphocytes. Do you mean lymphocytes? No, I don't think... No, because lymphocytes don't die after... Lymphocytes are what this is. So I don't know. What are the ones that engulf white blood cells that engulf pathogens and destroys them? It's just phagocytes. They are phagocytes. Those are the same. So there's phagocytes and lymphocytes. Phagocytes are the one that die, and lymphocytes are the one that do this antibiotic production. Neutrophils. That's what it is. Neutrophils, yes. Neutrophils are white blood cells. That's suicide, yes. Where was that? How have I missed that? I don't remember neutrophils. I know it's somewhere. I know we've talked, there's been talk of it somewhere, but I don't remember where. Yeah, my teacher taught it too, which is why I'm confused now. Here, we just Google neutrophils IB biology. Neutrophils, sorry. No, don't tell me the Google definition. I don't want to know the Google definition. That defeats the point of me Googling IB biology. God damn it. Okay, hold on. Neutrophils. It's not in the IB study guide, which defeats the neutrophil IB biology. It's in 6.3.5. Interesting. Okay, so there's different types of phagocytes. There's macrophages and there's neutrophils. Here, this is a crash course now. Okay, so neutrophils are the most abundant types of phagocytes, which basically will engulf the pathogen and kill them on the spot. Macrophages are the ones that bring them back to the helper T cells here. That's the difference. It's not mentioned in, my teacher did teach me, which tells you a lot about how much I remember of this. Let me also check BioNinja in case it's there, and then yeah, hold on, let's look at this. 6.3 phagocytes. Yeah, see, even this does not say the different types of phagocytes. So I think we should be okay if you don't. I think that's extra content. Yeah, that's extra content. You're okay. We're okay, guys. We're okay. I believe we're good. That's extra content. This neutrophils and basophils and all the different types of leukocytes is extra content. So I don't believe it's part of our syllabus. According to BioNinja, yes. And it's not in the here, just to verify if anything here. IB Biology Syllabus 2016. There you go. The guide. Let's look at the guide. The guide will tell me everything. If all else fails, just Google. Is the guide that detailed? Yes, it would tell you if you need to know the types of leukocytes. The guide tells you everything. If you go into the actual topic breakdowns, then it will tell you. If this freaking loads. Thank you. Dear God. Okay. So let's go down. Go down, 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 down. There we go. So topic 6. Yes. Topic 6. Let's look at this. Topic 5. Topic 6. There we go. 6.1. No, 6.3. 6.3. Understandings. Pathogens by phagocytic white blood cells. And production of antibodies by lymphocytes. So you don't need to know the specific types of leukocytes. We're good, guys. We're in the clear. We're in the clear. Okay, so now that we've fixed that problem, does everyone understand antibody production? Are we good? Are there questions that need to be answered? Just to recap, the first step is when the antigen is presented by the macrophage to the helper T cell. The antigen is then bound to the antibody in the inactive B cell. The inactive B cell then becomes activated by linking to the helper T cell. And then the activated B cell will divide to form plasma cells, which will then produce large amounts of antibody, which will then break down the antigen. And then memory cells will also be produced by B cells and T cells that are not activated. Good? I'm going to assume the silence to be no questions. But if there's any questions, you can... Oh, okay, yeah. The T cell... Yeah. Yes. It basically, what it does is identify the pathogen and then link to a complementary B cell. And then activate the B cell to then produce the plasma cell. Yes. Phagocyte, helper T cell, and B cell, yes. You need to know what a macrophage is, but... yeah. Basically, all you need to know about a macrophage is that it collects the antigen, but it does not kill it because it can't. So it just carries it to the T cells and B cells. Okay, I'm gonna keep going because there's only a little bit left, and then we'll do another break. So, antibodies and antigens. Did I skip something? No. Okay. So, antibodies look like this. There's this constant region, and then which is just these two darker gray lines. And then the variable region is the part where the other region comes in. So the constant region is the part of the molecule that aids the destruction of the pathogen. But then the tips of the variable region is what determines which pathogen binds to the antigen binding site, basically. So there are different versions of the constant region, which use different tactics to destroy the pathogen. So there's 5 different tactics that we need to know. The first one is making a pathogen more recognizable to phagocytes, so they're more readily engulfed. So it basically breaks them down to make it more recognizable, and then therefore just get engulfed by phagocytes and eaten through phagocytosis. And then the second is preventing viruses from docking to host cells. So it'll basically capture the virus before it's able to take over a host cell, and then will therefore act as an antibody. The third is neutralizing toxins produced by pathogens. So the constant region can basically, the pathogen will still stay here, but it'll basically stop the toxin production from it. The next is binding to the surface and bursting it, causing the formation of pores. Is it pores or spores? No, it's pores. So pathogens will usually have a surface, obviously, and then because these only bind to the actual antigen, the constant region can help it bind to the entire pathogen as a whole and the pathogen surface as a whole, and then therefore burst it and then cause pores, basically. So when you burst it, it'll basically become useless. And then the last one is sticking pathogens together, which is called agglutination, where two pathogens will be joined together and they can't enter host cells anymore because there's two cells joined together, but then phagocytes can also ingest them more easily. So this is the constant region, this is the variable region. The variable region is what determines what antigens will be bound, and they are the antigen binding sites, but the constant region is what allows our cells to destroy the pathogen. Okay, vaccinations. So vaccines contain antigens that trigger immunity to a disease without actually causing the disease to damage cells. So basically, vaccines are usually weakened or dead forms of pathogens, and once they're inserted into our body, our cells will be able to go through the entire process of antibody production, but it also ensures that there won't be any actual infection taking place, because usually antibody production will happen, but the person will still be infected. It could still be infected in any way, but the way vaccines work will basically ensure that the person is not affected. And then most vaccines just have the weakened forms of the pathogens, but some vaccines can contain the specific chemical that acts as the antigen. So the vaccine is either injected into the body or very rarely, but sometimes can be swallowed. The principle of vaccination is that antigens in the vaccine cause the production of the antibodies, and they basically go through the entire stages of antibody production. And sometimes there's 2 or more vaccines that are needed to stimulate the production of enough antibodies, which is why there's things known as booster shots. So COVID vaccines required booster shots because just one vaccination was not giving enough antibodies to prevent all the issues that came with the entire response to COVID. So usually when there's 2 or more vaccinations, the first encounter and the second encounter will result in an increased production of the antibodies, and it's not at the same level. Memory cells that are produced as a result of vaccines are usually persist to give long-term immunity, but this long-term immunity can vary a lot. So there's diseases or conditions such as chickenpox. Once you get chickenpox for once, the memory cells last basically for life, which means that you are not affected by chickenpox again, because the body is easily able to break the pathogen down before it affects your body. However, there's things like the flu, which they say you should get a flu shot every year because the memory cells that are produced as a result of the flu vaccination do not persist for longer than 1 year. But 1 year is still considered long-term immunity. It's just not long enough for the entire to last longer than a year. So yes, booster shots are needed to stimulate the production of more antibodies. It is also because of flu constantly mutating, but I'm just trying to give an example. So I could be wrong that it's flu, but it's just easier to understand when you have an example sometimes. It is because of multiple reasons. So flu constantly mutating could be one reason, but it could also just be that our memory cells just don't last that long in terms of flu. So, yeah. Okay, so the first infectious disease that was eradicated by vaccination was smallpox, and the person you need to know is Edward Jenner. So, the worldwide vaccination program was done in the 1960s and 1970s, with the last ever case occurred in 1977. But smallpox was also the first disease in which vaccine was tested on a human. So in 1796, Edward Jenner deliberately infected a 8-year-old boy with cowpox using pus from a blister of a milkmaid with that disease. So he basically saw that because there was a smallpox outbreak in his area, but then the milkmaids would never get that disease. But he also figured out that they all had cowpox instead. So then he deliberately infected an 8-year-old with cowpox because cowpox was not deadly, and then did an incision and tried to infect the boy with smallpox, but then found that he was immune to smallpox. So basically, the reason this worked is because cowpox is a less virulent disease caused by viruses similar enough to the smallpox for antibodies to be produced, but further away enough that it doesn't cause like a deathly reaction. Jenner then tested his procedure on 23 other people, including himself, and this method currently would be considered ethically unacceptable because they obviously involved a child that was really young, and the child would not understand the dangers and could not give informed consent, and then there was not enough tests done to figure out if there was any side effects. So this entire process, although it worked and it worked out pretty well for us, we would not be able to consider this an ethically valid process in today's day. Yeah, so ethics can be a really big thing. So there's a lot of ethics questions throughout the IB syllabus. I'd say most of them are associated with either experiments or GMOs. Okay, so this is the last bit, and then we'll do another break for like 10 minutes, we'll say. Okay, so monoclonal antibodies. So we've figured out a way to basically create antibodies, but in basically a limitless supply or large quantities, using a technique. So firstly, antigens that correspond to the desired antibody are injected into an animal. So usually it's mice. Those plasma cells will be produced within that mice through the whole process of antibody production, and then those plasma cells that produce that desired antibody will be extracted from the same animal, and then tumor cells separately that grow and divide endlessly are obtained from any culture of a tumor, and then that tumor and the plasma cells are fused together to produce hybridoma cells, so which will divide endlessly to produce a clone of one specific type of hybridoma, and that specific type will be of the plasma cells. So it'll just divide uncontrollably because that's how a tumor grows. So the hybridoma cells can then be cultured, and then their antibodies can be extracted and purified to form antibodies. So the antibodies produced by this method are called monoclonal because they're all produced from one clone of hybridoma cells, and they're all identical. One use of monoclonal antibodies is in pregnancy test kits. So the urine of pregnant women contain HCG, which is a protein that is secreted by the embryo in the woman and later by the placenta. So pregnancy test kits contain monoclonal antibodies to which HCG will bind, and that bond will cause a colored band to appear, which is why pregnancy tests have two lines usually, because one is the control line that's drawn on, and then the second one appears when HCG is present in the urine sample, and therefore the woman who produced the urine with the HCG is considered to be pregnant. Okay, that's pretty much all of 11.1. Yeah, go ahead, ask questions. I'm here to answer. Antigens are injected into an animal, yes. Here, there's a flow chart that I can put in if it helps you. So, there you go. So, what happens is this mouse is injected with an antigen. He forms plasma cells. B cells form plasma cells. Those plasma cells are taken from their spleen. Those those plasma cells that don't live very long are then fused with tumor cells that grow and divide endlessly. Those two fusions form hybridomas. Those hybridomas basically ensure a limitless supply of the antibody. Okay, so the way this enables pregnancy is monoclonal antibodies. Not this specific thing, but basically the antibodies that are produced here, the monoclonal antibodies that are produced here, bind to HCG, which is a protein. So that binding will cause a color to appear. That the HCG and the antibody bound together forms a colored line, and then that colored line proves that a person is pregnant. Yes, this is HL. Does that make sense? All the monoclonals will get tested, and the antibodies that are looked for, i.e. pregnancy, renal function, will be taken and put into the test. Yes. Yes. Okay. Cool. I'm gonna call this one